Hi everybody, I'm Brian Yandell, back again. I'm going to talk today about quantile-based permutation thresholds for QTL hotspots, and several words in there. We'll get through them as we go along. But basically we're looking at regions of the genome that show a high propensity to map for many traits to map to one position. So it's hence called a hotspot. Okay, and underlying this work is, it really goes back to Fisher. If you, you may or may not know it, but we have this Fisher's t-test, and the idea of using the normal distribution and and the t-distribution. Well, all of that stuff goes back to Fisher saying, if you have two groups and you're trying to compare them, and you shuffle the labels, you would destroy the group structure. And if you shuffle the labels a bunch of times and you recalculated the same statistic and looked at the distribution, it looks kind of like a normal distribution or kind of like a t-distribution. And so the t-distribution is a good approximation to the permutation distribution. And that got sort of flipped around, so people thought, oh, we should use a t-distribution all the time, and it's only when people started worrying about non-normal data that they went back to permutations. But the basis for it was this idea that if you permute the data, you remove the group different, the treatment assignment that you have, but you also are shuffling with respect to anything else that's involved that might not have been measured that's involved. So R.A. Fisher's a key to quite a number of things, and it's good to remember that genesis of the t-test. And so why study hotspots? Well, how do genotypes affect phenotypes? So genotypes are, and this, I was talking to someone just before we talked, the genotypes are DNA markers. That is, there's some piece of genetic material along the chromosome measured for an individual. And phenotypes are traits, some sort of observation measured on the individual. Okay. They might be clinical traits, they might be expression traits, they might be proteomic traits, where several people, there's some people here, I'm working with people who are trying to develop protein traits. And we want to look at the relationship between the phenotype and the genotype. Now, QTL hotspot is a genomic location affecting many traits. It was found early on when people started doing these genetical genomics or eQTL studies. And it was biologically interesting because there might be critical regulators in that area. That is, if you've got a huge signal cascade and everything in the signal cascade maps to one genomic position, then you might suppose that the top of that signal cascade, that gene, if there's the gene that created the molecular product for that, it may be located at that position, okay? Or something else that affects it may be located at that position. And so if you can get at that, then you start getting at the whole pathway rather than just at I found this thing mapped here, this thing mapped here. That gets old pretty fast, particularly when you're talking about thousands of traits or hundreds of traits. But the question comes up is, are these hotspots real? Or are they spurious? Are they just due to chance? So they could be coming up from correlation that's environmental correlation. Okay, so I showed this picture earlier. We have a clinical trait here and then a bunch of expression traits, and it looks like there's a bunch of things mapping to the spot. Can we make some sense of this? Okay. Now, one thing to note here, this is a view of the phenotypes, and at each position here we've done a t-test. We've actually done an f-test because there are three groups. It's an intercross. Well, we've done an f-test and we just connected the dots between them. So underlying this is the genetic material. So here's the genotypes where we haven't measured, and you see here's an individual that was black 6 and then became heterozygous. And then here's another individual who's heterozygous and became BTBR. We lined up all the individuals, just sorted them by their genotype, basically. And you see there's various rearrangements. So each line here is an individual. And because there's rearrangements, then as you move along the genome, your test changes a little bit because your groups change. Okay? That's the basis for that moving around. And you can take that and move up to, you saw a picture like this from Mark yesterday. I'm just showing it in black and white. Here we have about 100 traits looked over the genome. And you see these apparent hotspots here in chromosome 2, chromosome 17, and various other places. Here's chromosome 6. Okay, and I showed you this as well. Showed you that. Okay, so how large a hotspot is large? So there were a number of papers that came out in the early 2000s. You remember the 2000s? We're beyond that now. That proposed hotspots based on early EQTL studies. And Breitling et al. And Janssen basically provided an empirical logical basis for doing a test. So a hotspot, to define it now, is a count of traits above a LOD threshold at a particular location. And the LOD is the F-test, or rescaled likelihood ratio, with some adjustment for missing data. So basically what Janssen did was show how to do a permutation test to get at the null distribution, that is, if there's nothing genetic going on. And it's an extension of Churchill and Dirge's work, which was the QTL study that set up permutation-based thresholds for individual traits, which in itself was an extension of Fisher's permutation t-test, which I mentioned earlier. Okay. So the Churchill and Dirge approach, one trait, you're looking over the whole genome, and you calculate that LOD curve over the whole genome, find the maximum. Okay. Now you go back to your data, shuffle the labels. So you permute the single trait, you permute the genotypes relative to the phenotypes, find the maximum LOD, do that 1,000 times. So you've got your original data and you've got 1,000 permutations. I'll show a picture of this in a moment. And then you look at, I think I've got that here. Do I? No, I don't have that. Okay. So you end up with some sort of a distribution, and you find the 95% point, and that's your permutation threshold if you're using 5%. And then you want to identify interesting peaks, and then you've got to take some attention to controlled genome-wide error. Okay. This does control genome-wide error because you're looking over the whole genome when you do this permutation test. Okay. So here's the idea of the single permutation. So you have genotypes. So we've got the rows here are, think of this as a bunch of individuals, each row. You got a bunch of genotypes and you have a single phenotype. Okay. You go over the whole genome, you find the LOD, and then you find the maximum LOD. Then you shuffle and repeat. Okay. Now when you look at the, now we're going to make it a little more complicated. So now you've got, we have this, we're going to look at now the maximum count. Now we're going to look over multiple traits. Okay. So we got this 95% threshold that was the last two slides. They're usually something around 3 or 5, somewhere in that range. So, so then and let's say we got 1,000 traits or 10,000 traits we're looking at. So you find, at each position, you find how many traits are over the that 5% threshold, and you count that. So then you get a scan which looks like the LOD curve, but it's a scan of the count of traits, and I'll show you some of those in a few minutes. Okay. Now you take that and you permute in a certain way, and you repeat it 1,000 times. Okay, so you get the null distribution, but we have to be careful how we do this. Okay. So you just as you get a log curve across the whole genome, you can get a count across the whole genome. Let me show a picture here. Okay, here's one right here. Okay. So here we're looking over the whole genome, and we get a count of how many traits. I think there are about 6,000 traits here. This is the yeast genome data. We've got hotspots of size up to about 400, and how many traits are over a threshold of 7 in this case. Okay. So at this position on chromosome 2, there are about 400 traits over that. Chromosome 3, there's a parent hotspot here. Chromosome 8, we're right on the border there. 12, 14, 15. So at each position here, at each marker, we've counted the number of traits above single log threshold of 7. Okay. So now we have, what we have is again the same individuals, the same genotypes. And now instead of one phenotype, we have a collection of phenotypes. So this is a little broader, okay? We find the LOD at each locus for each phenotype over the whole genome, okay? So now we go from, we have the phenotypes and the positions, and we count the LODs at each locus that are over the threshold, over the LOD threshold. And we do that over the whole genome, and then we get a maximum of that count over the genome. So for this data, for the data itself, the maximum is something around over 400 here. But for permuted data, it's going to be something perhaps less. Okay. And here's what it turns out to be. So this is the permutation distribution for the hotspot size. And the, okay, so what we're looking at here is, so let me just explain this, because let's say we've got, we have here about 6,000 traits, and we got the whole genome. So we're looking at, across all 6,000 traits, what's the maximum hotspot size across the whole genome? Okay. And we're looking at hotspot size of size 1. So what's the chance that there is anything anywhere in the genome that's significant? Okay, hotspot of size 1 doesn't really make sense, but if you think about it as we got 6,000 traits, what's the chance that there's anything interesting here? Anywhere in the genome, any genetic signal. Okay. So you would take the, you're going to look at of a 1 trait threshold. Okay. I'm confused here because the label doesn't agree with what I'm got in the figure. Okay, so this is a threshold. Let's see, 82 is a threshold when we use a 95% threshold when we use a single trait threshold of 3.41. Okay. Now, if we use a okay, wait a minute. I have to back up here a moment. Okay, so if we use a trait threshold, a single QTL trait threshold of 3.41, which is what we would get from a 95% permutation test with a Churchill dirge, then we would find and we did 1,000 samples, we would end up with a distribution something like this. And the 95% threshold is at 82. Okay. If we instead used a threshold corresponding to a single trait, where we say what's the chance of having any greater than anything at all, we'd be using a threshold much larger than this. Okay, so let's see if I've got that. I somehow got mixed up here. I'm sorry. Okay. Okay, so let me go on here. Okay. So here's some more detail about the permutation test. For the original data set and each permuted data set, you're going to set the single trait threshold, and we count the number of traits. So I've said this already. Find the count above that and conclude the original data counts are above the threshold or real. Okay. So here's a graphic of what's going on here. So here we have the genetic data, and here we have the expression data. And again, each row here corresponds to an individual. This is original data here, and the key, this is the key point to understand in this about these hotspots. So we have all this genetic data for an individual. We have all this phenotype data, all this expression data for an individual. What we want to do is break the connection between the genotype and the phenotype and shuffle the phenotypes relative to the genotype. But at the same time, we want to keep the genetic relationship here, which has to do with linkage across all these markers. And we also want to keep the correlation among all the phenotypes. So we keep all the phenotypes together. We keep the rows together, but we separate the red-green from the yellow-blue, and we permute them. So in the bottom, we permuted the genotypes. You could permute the phenotypes instead, but in the bottom, we're just permuting the genotypes. And now you have a different genotype for the first individual, but it has the same phenotype it had before. So you've shuffled the relationship between those two. Now, what will happen if there's no genetic signal is that the hotspots will just move around. They'll move from one position to another, but they will stay as hotspots. If it's really a genetic signal, then you'll actually break up the relationship, and the hotspots will disappear. Okay. So you're breaking the correlation between the marker genotypes and the trait phenotypes, but you're preserving the correlation among the traits themselves. Okay. Now, one of the things that we found when we use this for our diabetes data is that we have a particular insulin trait on chromosome 19. And we looked at the hotspot, it wasn't quite significant. So what do you do? Well, we could say, well, maybe we should just sort of adjust things a little bit, but you want to be careful about doing that. You try to set up an objective criteria and follow that criteria through to the end. So what we realized is that maybe our criteria is flawed in some way. So as we thought about it, we realized that what we were doing was setting a threshold regardless of the strength of the signal for each of our traits. So suppose you had, so here we had, in the example I was showing you, we have a cutoff of 80 traits. Okay. But suppose there are 40 traits that show really strong signals. Now that could correspond to a signal cascade, which is really important, but it would fall under the radar if your threshold is 80. You would never detect it. You'd say that's not significant, we can't use it. So if we instead looked at not just how many traits fall over a threshold, but we took account of the quality of the traits that were in there, like how strong was their signal. So finding large hotspots with many things in them may be important or maybe not. If you get 1,000 traits that all have a very small signal, it's sort of like what Steve was talking about with the neighborhoods. If you have 1,000 traits that have a very small signal, is that going to be more interesting to you than 50 traits that have a really strong signal all at the same position? Maybe you want to look for both of those. Okay. So we set up a sliding threshold approach, which will get at both of those. Okay. So we got to rethinking the approach. So the breeding at all approach depends highly on the initial threshold that you set. And and that's based on a single trait. Okay, that's okay. We can still work with that. But we're really interested in working with multiple correlated traits. So what one could do is go back and say, well, let's develop a method that deals with multiple correlated traits. But that gets kind of complicated. So what we decided to do instead was to think about using a single trait but adjusting the threshold. Okay. So I think this is getting to the point that I jumbled up earlier, so let me try again. So we want to control the genome-wide error rate for a hotspot, and hotspots of different sizes. So what's the chance of detecting at least one spurious hotspot of some size n or larger? That's the question we want to ask, and we want to come up with a criteria for that. Now, when n equals 1, this is the special case, we're looking at the chance of detecting at least one peak above a threshold across all traits and the whole genome. Okay. So we use permutation null distribution of maximum LOD scores for all transcripts in all genome locations. Okay. So we're going to just throw all the transcripts in all locations and then just permute them. Okay. So here's what happens. So that other one that was mislabeled threw me off here. So this is what goes on when you look at hotspots. So we're going to look at hotspot architecture using a multiple trait genome-wise error threshold. So if we use a genome-wise threshold where we protect against anything happening with any of the traits, we have to be more conservative. So instead of using a threshold of 3 or 4, we have to use a higher threshold. It turns out it's about double. It's 7. So this is our conservative threshold, saying anything above there is going to be significant no matter where we're looking and no matter what trait. We set ahead how many traits we're looking at. It's about 6,000. Okay. And so here what we see, the threshold is down here, and we're seeing that we have something significant across many parts of the genome. Okay. So, but the counts are lower. Before, the counts here were 400, hotspots of size 400 or a couple hundred. Now the hotspots the highest hotspot is a little over 100. So here we have 100 traits, 100 plus traits that have LOD score over 7. Now there are 400 traits that have LOD score over 3.5, but only 100 that have LOD score over 7. Okay. So these are presumably the stronger signals. Okay. And then we've got these other signals coming up, and this chromosome 8 is still showing up here, is now showing up as significant. So when we take a much more conservative threshold, we end up picking up different kinds of hotspots. And you might have a situation where some hotspot that was picked up before doesn't show up at this point. Okay. So now, well, we looked at 1, we could look at 10, we could look at 20, we could look at 50, and we could come up with different sort of thresholds for different hotspots coming up. I just have one threshold shown here, but for hotspots with a of 10 traits, 20 traits, 10 traits, 20 traits, or 50 traits. Okay, using the threshold. Okay, so so we're now thinking about it as like a quantile. So when you think about 10 traits, let's say 10 out of 6,000 traits. So 10 over 6,000 would be the quantile. So you're looking at the tail of the distribution of the LOD scores. So what I'm saying here is that the top 10 LOD scores. Okay. So quantiles, so if we're looking at the maximum LOD quantile by trait count, okay, so if we look at counts of 10, we get 24 on chromosome 3. These are the heights of the peaks. And as you look at higher counts, they go down. Okay. Okay. So now we're going to revisit what this permutation test is about. We have the same genotypes, they have the same phenotypes. We calculate the LOD at each position. And then instead of finding for one threshold, we actually look at a bunch of different thresholds. We actually look at, instead of looking at the thresholds, we look at the n largest LOD scores that are found there. Okay. And then we find these quantiles. So we're looking at the distribution of the tail of the LOD scores across these correlated traits at each position. And then we maximize that over the whole genome. Okay. Okay, so when we can think about getting a spurious hotspot at particular locations, some of these are going to be small hotspots with higher LODs, and some of them are going to be large with smaller LODs. And the threshold that we use is now a sliding threshold that depends on the hotspot size. So instead of setting a threshold on the size of the LOD and then seeing what size hotspots we do, we say, I want to look at hotspots of a certain size, and what would the threshold need to be for me to pick up that size hotspot? And so we end up with some sort of a distribution here of the possible LOD thresholds and the hotspot size. So with a small hotspot, you need a larger threshold. And these are, this is 5% and 1%. Excuse me, let's see. Yeah. And there's a sort of minimum level that we want to do right here. This black line here is a minimal level which corresponds to that 82 we had before. So we never want to go below the single trait threshold, which in this case was about 3.4. So if we were looking, we could actually look at much larger hotspots with a smaller threshold, but then we wouldn't trust the individual traits. So we actually cut this off right here. But you see you have a sliding scale, and now the threshold is determined by the hotspot size as opposed to the other way around. Okay. So now we end up with an architecture here of hotspot. Let's see. This is again what I showed before with the most extreme, the threshold for the most extreme LOD score. And now, and this is the one I showed before, although now it's got the lines on it, for the hotspot architecture for different sized hotspots. Hotspots size 10, 20, or 50. And now here, what we have is this significance profile for hotspots. We're basically saying there are hotspots all over the genome, but the hotspot, but some of these are really small hotspots. So there's a region here on chromosome 4 where hotspots are only 5 traits. There's only 5 traits in here. Here's chromosome 8, which didn't quite hit a threshold before, but we're seeing 75 traits that are significant there. We knocked it off at the top here just to keep it on the screen. But what's happening is we've got a sliding scale here. So for a hotspot of size 5, you need we're only considering traits that are 5, the LODs are 5 or higher. For hotspot of size 75 here, they can be 3.5. And the maximum is 3.45. So in this way, we can get at a more nuanced look at hotspots. So what happens here is that the large hotspots that we had before are still significant. They remain significant. The traits that have really large ones, those could be hub genes. Those would be a place where you start looking at hub genes. Although in this case, there are a lot of them to work with. So they would be you'd want to look at this in connection with other lists that you have. Smaller hotspots have large LODs, such as the one I showed on chromosome 8, and it gives you instead of setting a hard and fast rule, it gives you a subjective thing. So if you are actually interested in chromosome 5 for other reasons, then you would never look at it based on the hotspot analysis. of a fixed threshold here, but you could look at it here and say, well, there are actually a number of traits here that have LODs of 4 or more, and that's significant. So we have 30 traits or so with LODs of 4 or more and that makes a significant hotspot. And you can defend that, and you can go ahead and do follow-up, look at that subset at those multiple traits. Okay.
I have a question.
Yeah.
So the y-axis, this hotspot number, that's the number of traits.
This is the number of traits in a hotspot. Yeah.
That are correlated, or is it all combination?
Well, they're correlated. But there were on chromosome 8, there were about 75 traits that mapped to chromosome 8 with LOD scores above 3.5. So those traits are probably correlated. They probably have a strong genetic correlation among them.
And you're not specifying any explicit linkage or linkage disequilibrium.
Well, the linkage disequilibrium is reflected in the mapping. So
you're using this sliding threshold, right?
yeah, the sliding threshold is basically this threshold over here. That as you look at larger hotspots, you can be more relaxed on the size of the LOD score, of the maximum LOD score.
What if it's a larger haplotype block? How does that affect the threshold?
The larger what?
A larger haploblock. So there are spots that have like chromosome 6 in humans. There's a lot of polymorphisms.
Yeah.
Right. So there's not a lot of linkage. There's a lot of.
Well, if you have very low linkage disequilibrium, then you're going to get a choppier picture here. So this is in yeast, and you do get some bouncing around here. In mice, it's a much smoother picture. So this doesn't really have to do with haplotype blocks.
Well, what's the relationship between the sliding threshold that you use and the size of a haplotype block?
There's no relationship. So that's a different concept. It's orthogonal to what we're talking about here. So so what we're doing is at each position on the genome, we're doing a t-test based on a marker at that position, okay, across all the traits. Okay. So which traits have the most significant signal at that position? You count how many have have signals above some sort of a threshold, okay? And then we're letting that threshold change, and we basically start at the bottom here and say, all right, how large can the threshold be in order to count things as significant? Okay.
But in regions of dense polymorphism,
yes,
there's more opportunity for you to get a false positive, right? Because you're testing more sites at that location.
I think this.
Yeah, I'm not sure.
It's not
Yeah.
based on. It is not a association test. It's
Yeah.
a linking test. So what really counts is not the our linking is equal to the linking. Those are two different things.
yeah.
So the permutation, they take the number of markers into account. So I don't think there is a big.
You want to help me out, Steve?
I would say let's discuss it later when we have a cookie and some coffee.
Yeah.
We will all
Yeah. I think you're mixing two different ideas together, and that's part of the problem here. There are issues that you could get that if you have very little linkage disequilibrium, then you can end up with a very choppy picture. And in fact, one thing we did was smooth things out to try to address some of those issues. So that is one thing you can do. Okay, so I want to see, where are we on time here? Okay, I want to just shift gears a little bit here in this talk and talk about that what I just showed you had to do with a yeast study where there are about 6,000 traits and 250 markers, 1,000 permutations. And so there are basically 10 to the 10 different models that are being fit here, okay, on these 120 individuals. In our mouse study, we had 500 individuals, 30,000 traits, 30,000, 40,000, depending on how you count them, 6 tissues, 2,000 markers, 1,000 permutations again. You're at 10 to the 13th, so it's 1,000 times bigger. So the yeast study we could do in a day or so. So this would take 3 years. So we realized we had to do something a little bit different. So this has to do with scaling up, and it's something that we're all having to deal with in different settings. There's just the big data issues that are coming at us. So there's a lot of issues in terms of the quantity of data and the high throughput issues. I'm just going to talk about what we did in this setting. So what I did was break this up into 1,000 different jobs and send, or actually 10,000 different jobs, send them out to a bunch of different computers, and then pull the stuff back in. And I got it done in half a day. So this is just showing the scheduling of all of the jobs that got done. So this is scheduling, this is actually running. But this worked. We took about 11% of the time on the open science grid for a particular day, but most of the work was being done by engineers. They do this every day. They're using 75-80%. So they're doing 10,000 hours, and I only did 4,000 hours on the computer. Okay. So anyway, because we're doing permutations, each permutation is independent. You can send each permutation off to separate machines, and you're doing a bunch of different traits. You can do each trait separately. So you can effectively send each trait or a set of traits off to one machine, break it up into a large number of different small tasks, and then just gather them together at the end. Okay. So when we do that, then we can end up with ways to sort of look more carefully at the threshold. So this is with the mouse data. So let's see, here's the 5% threshold. 5% threshold was about 3.9 something, which would correspond to hotspot of about size 400. So for the mouse data, using, and this is using a 5 centimorgan window related to some of the issues that were just raised a few minutes ago, we had hotspot size about 4,000. Without smoothing, it's about the same. 400. So anything above 400 traits, the same position, is significant. Okay. And so here we got a particular chromosome, and we've got males and females combined is the black set. The blue set is the males only, and the red set is the female. So you conclude from this that there's a hotspot, that the hotspot is largely for males, that it's located right here. This is where you should do your follow-up work. Okay. So that would be the end of the story. But if we take the sliding scale, and we're basically saying we're turning the axis around. Now we've got hotspots on the lower axis here. And here's the one we had before, 391. But as we look at smaller hotspots, we allow for a larger threshold. And the largest threshold is about 8 or 9. So for a single trait. So anything above 9 for any trait is going to be significant, considered significant. And now we have the sliding threshold. We have black as combined male and female, blue as males. And you see that again, we got the same position as the highest one, but we also have hotspots. We have sort of a much larger hotspot over this, this whole region. There are just fewer traits out at this other side. And again, the scale is sliding here. The other thing is these the females here are showing significant hotspots as well. They're just smaller hotspots with a stronger signal. So this would be missed entirely if without this sliding scale method. Okay. So there's follow-up studies that could be done on this in terms of assessing the power of the test. We've done some of that in publication. The key thing is drilling in on hotspots and trying to find out what they mean. Okay. So a hotspot, you might think that a hotspot would be a bunch of traits that are all highly correlated with each other. So remember that there's a couple things going on in these hotspots. One thing is that because of, for instance, in the mice, because of the linkage disequilibrium, there could actually be multiple drivers and multiple cascades that are tightly linked together, and we're seeing these basically overlap of these different signal cascades. And each signal cascade could be slightly different, and so you could have subsets that are correlated tightly with themselves, and other subsets, and they're not tightly correlated with each other. So this is also something you see with the modules. If you have a large module, you can have subsets of the module which have different characteristics. So so one of the things that we've been following up in is looking at causal agents, and Elias will be talking about some of that. And then also, if you get a small enough set, building causal networks, and again, Elias will talk about that. And there's really a challenge with some of these large hotspots because there's just so much going on. It's hard to tell what's going on. So even though this, you can find this something significant, there are a lot of genes in this region, a lot of genes to deal with. So it's not clear. This is, as we were talking about earlier, this is one of the shortcomings of doing QTL studies with experimental crosses, that you end up with, you still end up with a large list of tens or hundreds of traits to work with. Okay. And then there's some references, and that's the stop for that. So let's take a break now, and then we'll come back. I'll do a demo using yeast data and walk you through the various steps of the, well, I'll tell you about it when we come back.